Let's start the trip together. And actually, that's the wrong one, the other one. So how are you doing? How are you?
You pressed for
What is forward, the green?
forward the screen.
Okay.
Yeah, and the red is the laser. The one that looks
Okay.
has a laser symbol.
Right, thank you. Okay, great. So you see the title of the lecture is From Nanotechnology to Nanodrugs, Our Doxil Experience. And basically, you know, I hesitate how to call this medicine for a long time. And when Doxil was about 10 years old, the New York Times have a small article about it, and it said it's the first nanomedicine to be developed. If New York Times take it seriously, I should join. And since then, I really keep telling it the same. Actually, you know, we liposomologists, for a long time, we were not considered nanotechnologists. But I'll go into this point later. I think because we represent some old work and the nanotechnologists come with new particles, and we'll see what's the difference between them. So the first thing I want to say, we all know that there are many modern treatment of drugs. The most published one, the most people show a lot of interest are the antibody and the novel antibodies. But what most of the people don't know is if you give these antibodies without any what called conventional chemotherapy, they will not work. You need the support. And actually, most of the work done by the first-line drug, by three very good chemotherapy, the cisplatin, Taxol, and doxorubicin, so it's about four, and cisplatin. These four drugs are the leading drugs to which most of the cancer patients really respond well. We know all the downside of this, and the question we have, if we have to live with all this chemotherapy for a long time because of reasons specified here, and other many reasons like resistance that I don't mention here, then how can we do it in a better way so the patient will benefit more, therapeutic index will be much better, and the chances actually to get help from this modern, more up-to-date system will be much more beneficial to the patient. So that's actually what I said, how can we improve it, and that's what I'm going to show you. So actually, what we're trying to do is actually to improve the therapeutic index, but getting less drug into irrelevant tissue, thereby lowering the toxicity, and on the other hand, focusing and trying to get more drug accumulating into the disease site, like in the case of here, if we have a cancer of the lung, for example, or can be inflammation, it's working the same way. How can we concentrate more drug here? There are a few ways to do it, and I'm not going to talk about them. One can be by a default of a promoter, which will be tumor specific and will activate a certain drug in a certain way. So if you reach the cells which are healthy, nothing will happen. The other is active targeting to some cell surface specific antigen or other ligand, and the other is targeting what I called hitchhiking on the disease, which is targeting due to the specific property of the vascular heart, which really occur in most tumors and as well in most cases of inflammation. And it seems to work now very well in more than 90% of the tumor that it was tested. There are some exceptions. The main one is pancreatic cancer, and maybe this is one of the reasons that pancreatic cancer is very difficult to deal with the drugs. So we're coming here to, first of all, to the old and very attractive utopia that was put ahead of us by Paul Ehrlich, and we're trying to see if we can really come close to it. And basically, all the Doxil idea is based on this concept, and I'll try to convince you that we are coming very close to it now. So the first thing, I'm not going to talk about cancer biology, because I know very little about it, and the second, we don't have time. But what I want to talk is about another aspect of cancer, which many of the physicians and the people tend to ignore, and this is cancer physics. Cancer physics is a field that actually is pushed very hardly by Jen from MIT. It's a very important aspect in which inside the tumor you have very high pressure, and this pressure actually, this pressure that we see here prevent diffusion toward the tumor from the vascular artery. So while in the normal tissue, this is responsible for the way most drugs reaching this site. So I think this is a very important aspect that actually makes the chemo even less efficient than it should be if there was no such effect. And you can see all this pressure down here. This is slide taken from a Jane paper in Scientific American. Now, the other concept we want to really take advantage of, and this was the hitchhiking of the disease that I said, is the EPR effect based on two basic principles. If we look on the vascular artery in the normal tissue, we can see a very tight membrane of the blood vessels. Particle cannot leak out. All the drugs, all the molecules are really go out by diffusion, and inside this tissue there is a very effective lymphatic drainage that if particles for some reason present, they will be removed by the lymphatic drainage. EPR effect that was discovered in 1986 by Madea and Matsumura, actually, two Japanese researchers. One of them is still working. Matsumura was the teacher and Madea was the student. And what actually happening in the tumor, because of the uniqueness of the blood vessels there, without going into the biology again, it's a porous blood vessels. So particles at the size range of 100, 150 can really slowly but surely leak out to a very large extent. And for reasons that, again, not clear, this tissue lack a drainage system, so the particles will remain here for a very long time. This is basically the EPR effect, of which we actually found some paper in 1987 and start working on it right away, and it proved that it's working very, very well, and I'll show you some data in human later. Actually, most of the data I'll show are human data. These are liposomes. I don't think we have to say much about it, but it is a very versatile system. It's based on a lipid bilayer, and I think today is one of the best drug delivery system. One of the reasons is that we know so much about it that we don't know about the other systems. So it gives us a huge advantage. It has a membrane and a milieu interne in which both this part we can actually include or encapsulate drug, so we can deal with hydrophobic drug and with very hydrophilic drug. Both are very good candidates. There is a very large repertoire of liposome. Today I'll deal only with this up to the size of about 100 nanometer, which we call the nanoliposomes. It can be either smaller or larger. This really has a reason why the 100 nanometer is a kind of a limit, and that's because when here we have only one line, but it's two bilayers. When we have below 100 nanometer, the curvature of the two leaflet is different. When we're reaching 100, it becomes about the same, and frustration that's in the system completely disappears. So it has a physical significance. You see other type of liposome we are using. This is used for the talk I'll give tomorrow, or this afternoon, sorry, and this we're using, for example, for local delivery of local anesthetics, and this we use for vaccination, but I'm not going to talk about it today. Now, liposome has different function. We are going to talk today about liposome that circulates for a very long time, and if they're small enough, they will actually leak or be released at the site of the tumor. They have a very long circulation time. I'll show you in human, up to about T-half of about 100 hours, which is about, now to our taste, too long. They are not taken up of the RES, by the RES, and that's why they are so long. This type I'm not going to talk today.